Welcome to this week's course of Intelligent Healthcare and its applications. So this week we will move on to the pathology image analysis. So following the previous week, I think you should have some insights into convolutional neural network and how this innovation neural network can be applied to the imaging analysis. So in medical fields, that there are lots of medical images, and pathology is one of the medical specialties that are heavily based on the visual identification of the pathology patterns of these imaging slides. So this week I will show you the trend of the digital pathology, and in the workshop I will show you how to apply CNN model to classify the cancer or tumor pathology images versus non-tumor parts of the images. So I hope that based on this week's course that you should be able to work on the case presentation too, using the codes I attached to this week's lectures. Okay, so let's take a look at this common scenario that's in the hospital. So imagine that you are doctors, and then there was a patient coming to your clinics, and then you are going to evaluate these patients. So this is a 62 years old male patient, and his chief complaint was a cough with bloody content. Okay, so you are trying to ask about his past history, but none of them are significant. So maybe he has no hypertension, has no diabetes, has no other previous diagnosis of the systemic illness. So the only personal history you found is that he has smoking 2 packs per day for 40 years. Okay, so that was the history you collected in the clinics. So imagine that if you are doctors and what you are going to do in the next step. So you probably be wondering that, okay, so because his chief complaint is cough with bloody content, and plus he has a smoking history, so you are wondering that if he has a problem in his lung, then of course you need to do some physical examinations. You need to use the stethoscopes to make sure there's no if there's any abnormal breathing sounds, or you need to examine the other parts of the body, including the heart, including the abdomen, the bowel sounds, and the other physical examinations. So, but based on this common scenario, then we could that probably the highest priority is to do some imaging study. So if you are going to do imaging study, that maybe the first choice is the chest X-ray. Okay, so that is the results of the chest X-ray. And in the chest X-ray, then usually we will try to evaluate the several things. The first thing is that you need to look at the lung fields and the heart, and also the diaphragm, and also the trachea, and also the ribs. Okay, so maybe the first thing is that you need to scan the chest field, the lung fields, to see if there are any abnormal findings. For example, if there's a effusion in the lung, then usually the fluid will accumulate in this region. So if you see fluid accumulate in this region, we call the CP angle, then probably you will think that, okay, this patient may have a lung effusion, pleural effusions, okay? Then you will look at the lung field to see if there's any patch. Okay, so just give this example to you that to show you, although the chest X-ray is a very common imaging studies in the hospital, however, the reading of chest X-ray is still very challenging. So every medical students will, oftentimes we will have a course of the chest X-ray reading when is in like an intern training period, and so that is almost the basic skills for every doctors. However, because just like I said, the chest X-ray just relies on the penetration of X-ray through our chest, the part of the body, and you can imagine that the X-ray beams need to penetrate through different types of tissues. So you need to build up a 3D anatomical view in your brain just based on these two-dimensional images. So it is very challenging actually. So in this case, you see that okay, there's no other abnormal findings in this part of lung fields. These are the blood vessels. So this is not abnormal findings. And probably you also look at the heart. Okay, the heart is within the normal limit. So I use the term within the normal limit to, this is a very common term in the hospital because we usually do not say it is normal, but we say it is within normal limit. Okay. So the other part you can see there's a altar, there's a shadow of the altar just coming through this way and going down to the abdomen part of the body, and the diaphragm seems okay, and there is no fractures in the bone. So if you are the medical students, or if you are not a radiologist, you probably will think that this is the normal chest X-ray. But however, I would say that chest X-ray is not always the final imaging tools that you will use. So in that case, because your patient is coughing with bloody content, then but it's also very challenging to see anything in this chest X-ray. So you might want to do the computed tomograph, which is called the CT scanning. So the CT scanning is also based on the X-ray, but instead of just using X-ray penetrate through one direction, the computed tomograph will use X-ray to penetrate through different directions to reconstruct the body images from the result. So the resolution of the CT is much higher, and it has more insights into the three-dimensional anatomical representation of your body. So it's very easy to see that in this section, you see there's a tumor attached to the pleura and grows into the lung organ. So this is definitely an abnormal findings and need to identify whether or not it is a tumor or it is a benign tumor or it is the cancer. So based on these CT images, let's go back to this chest X-ray. You'll probably see that, okay, actually there is a very just not obvious shadow here, vague shadow here that actually is corresponding to the CT images. So that's why I said that actually the reading chest X-ray is very challenging, because when a tumor is located in a region that has the multiple tissues in the same direction in layers, then it's very easy to miss that abnormal findings. So this is actually an abnormal chest X-ray, but because based on CT we found that there's a tumor here, then we go back to the chest X-ray. Okay, this is very hard to find tumor here. And I would say that a very experienced radiologist can see these things, but if you are medical students or if you are doctors that is not have specialty in radiology, then probably we have a very high chance to miss this tumor. Okay, but anyway, based on the CT images, then we find these tumors, and the next thing is that we need to know whether this tumor is benign or the cancerous things. Okay, so how to justify this tumor? So the only thing we can do is that we need to do a biopsy, which means that we need to extract a small amount of tissue from this tumor. Then the thing is that we need to do a biopsy either using a surgical procedure, or you can do a needle biopsy. That is just penetrate using a needle, just penetrate through the pleura and to get some tissue. But in this case, okay, the doctor decides to do the operations, the surgical operation, to remove the tumor and also identify what's the nature of the tumor. Okay, and the doctor successfully extract a small amount of tissue from the tumor, and the next thing is to ask pathologist to see whether or not this is the tumor or benign or cancerous tumor. So the pathology was called up into the hospital. Then the pathologist need to process these tissues, and because the patient is still on the surgical table, so we need to do things very quickly because the patient is still under anesthesia and cannot wait for a long time. So the pathologist just put this tissue in this frozen machine. Then this is frozen by the liquid nitrogen, and you can see that tissue will be frozen, and you can just use the very sharp knife to cut into the very small, very thin pieces of the tissue. And these thin layers of the tissue will drop onto this part, and you can use the glass to just attach to this tissue, and you can just make a pathology glass. Okay, so this is the one slide of the pathology tissue, the sample. Then the next thing is that we need to visualize the nuclei in this tissue. So one way to differentiate the nucleus from the cell plasmid is the HE stain, which is the abbreviation for hematoxylin and allicin stain. So if you are interested in this procedure, then you can look up the internet about the HE stain, then you will understand this procedure. But basically, the nucleus will be labeled as the purple colors, and the plasma will be labeled as the purple and red colors. Okay. So now, because we already prepared the pathology slides, then the next thing is that the surgeon and pathologist will work together to look at these slides to determine that if this is the tumor or benign or cancerous tumor. So you can see that if this is tumor, then we need to remove entirely about this tumor, and so before we can close our surgical procedure. However, and if this is benign, you still need to remove the tumor, but you don't have to examine the surrounding tissues. For example, if the tumor is benign, then you just can remove these tumor parts. But if the tumor is the cancerous, then you need to examine if the tumor has invasion into the surrounding lymph nodes or surrounding tissues. Then you need to remove the more. For example, if you probably need to remove the one section of the lung to make sure there was no tumor cells in the other parts of the body. Okay, so that is the daily routine occurred in the hospital. Okay, so this is the final pathology slides. You see in this frozen tissue. So you can see that it is definitely an abnormal tissue because this is squamous cell carcinoma. There's lots of the squamous cell grows into the very random patterns, and this is definitely not normal. And compared to a normal squamous tissue, you can see that the squamous tissue is kind of the cells that grows from the base membrane and it gradually characterized in the surface. So you can see that in the normal tissue, the squamous cell has quite a regular alignment, and it has a very clear boundary. And compared to a squamous carcinoma, you can see that it just grows everywhere without boundaries. So that is a very visual patterns that you can identify between the cancer and the normal tissue. So let's review the whole process, okay? So when patient comes to your clinics, then you need to do something to do imaging studies. Then in this case, you find that, okay, this is there's a tumor you found in the lung. Then you do the surgical procedure to extract a small part of the tissue to make sure this is whether it is the benign tumor or the cancerous tumor. Then in the new era of the pathology that you probably don't need microscope. You can use the slide scanner to scan all the tissue in the high detail, and you can visualize the whole slide scans on your computer screen. Then you can just type a report based on the computer scanning. So that is one of the revolution that maybe we can digitize everything, including the pathology slides. So this is a core of the digital pathology. But imagine that if you can use the machine learning or AI to identify these things, that would be more powerful because if you are doctors and imagine that you are working very late in the midnight, then you probably will miss a tumor. Suppose that the tumor is very small, it just hide into a small location of this a particular location of the slides. Then you just scan the whole things, but you just miss that part. So that is why we probably will need AI to prevent doctors from missing diagnosis of the cancer or other abnormal findings. Okay, so this is one of the figure that is showing the 10 applications of AI in healthcare. So, like I said, one of the major research and application is focused on the AI-assisted medical diagnosis, but do not and medical imaging analysis. But do not forget that there are other parts of the AI you can also apply in a smart healthcare, including the AI-assisted robotic surgery and virtual nursing assistance, drug discovery, clinical trials, fraud detections, medical risk prediction, which is also very important, automated workflow assistance, and medical data security. So these are all important areas of the AI application in medical fields, but so far the majority of the focus has been on the medical imaging analysis. Okay, so let me give you just a mini course that how pathologists can identify the tumor, the benign and cancer. So this is the one of the pathology images from the liver. So we call the liver images contains the hepatic cells, and that is where the cells can to produce the bile. Okay, so I just show this one, and you can see this one. Probably you have already have some insights that okay, these two are different. In this one, the dark purple color means the nucleus, and the light red or light red part means the plasma of the cell. Okay, so you can see that in this one, it has still kind of regular cell arrangement, but in this one, the arrangement seems to be more random, and you can also see that the nucleus part is more dark, means that it has heavy activities in the DNA replications, and there are lots of the chromosome, because then so you can see darker colors by the alzine. Okay, so based on these two liver pathology slides, I just want to show you one of the visual rules that can identify the benign and tumor. But however, sometimes it's very difficult to see just to differentiate the tumor, the cancer and benign just based on the visual identification. So in that case, then you need molecular pathology, which means that you can use the immune markers that to identify the special types of the antibodies and antigens then on the cell membranes, and you can see some cancer has a specific antigens that you can identify with the immunohistochemistry profiles. So this is one of the major breakthrough of the pathology, because for molecular pathology, you relied on the biomarkers. Then instead of just looking at the morphology of the cells. So, but just let me just put several things together into this slide. So there are 4 liver pathology images, A, B, C, D. Then only one of these is the cancer cells. So which one is the cancer? Okay, so probably you already know the answer, because in A, B, and D, there are the cell arrangement is still okay, but in the C it seems very regular. So this one is the not is a cancer, is a hepatoma images, which is the liver tumor, but the other things are normal. Okay, so how to differentiate this tumor and normal is a challenge, but I'll just show you in these slides. then you can just use some imaging analysis to identify the nucleus of these pathology images. Then you can identify the numbers of the nucleus. You can also identify the location of this nucleus, and you can quantify these things with computer programs. So this is actually one of the study I conduct about 10 years ago. That at that time I just write a program myself to identify the nucleus in these images, and we can quantify the nuclear morphology just based on nuclear density, nuclear area, parameter, and nuclear diameter, and roundness, and ellipse, other characteristics of the nuclear morphology. So at that time, you can see that I use this particular mathematical methods, which is called the Delaunay triangulation, to identify the randomness of this nucleus. So let me go back to this one. I just want to show you that the pathologists actually have 4 principles to differentiate the tumor in benign and cancer. So these 4 principles is that for the cancer cells, it usually has a larger nucleus because it has more activities in DNA replications in the cancer cells. It has larger, and because it has more DNA replications and more chromosome activities, it will be more darker in color. So the first one is the bigger in cancer nucleus, and the second rule is the darker color in the cancer nucleus. Then the third rule is more diverse, the morphology. So it has different shapes in nucleus. The shape is not regular. And the fourth principle is that the arrangement of this nucleus is very irregular. So let me repeat these 4 principles. The bigger the size of the nucleus, and the darkness of the nucleus, and the shape of the nucleus, and the arrangement of the nucleus. So these 4 principles is actually the kind of the rules that are used by pathologists to differentiate benign and cancer. Okay, so in this study, I also used the mathematical calculation to differentiate the tumor and the normal cells. Okay, so it just try to measure the randomness of the nucleus locations. So based on these examples, I just want to show you that in the pathology image analysis, the first thing you need to do is digitization. And the second thing is you need to do the digitalization, which means that you need to analyze these data using some algorithms. And one of the powerful algorithms in the modern computer science is the artificial intelligence or machine learning. So that's I want to show you these 2 steps. So in the digital pathology, the first thing is that you need to digitize everything of the pathology slides. So one of the advantage of the digitizing these slides is that any pathology slides the color will be faded in the long, just as time goes, pass on. So imagine that in the hospital, we cannot discard any pathology slides because that slides is parts of the medical records. So probably no one will want to dump these slides just because it's very old. But imagine that if it is a very big hospital, just for example, like the National Taiwan University Hospital or the Taipei Veteran General Hospital, the storage space will be need to have a large storage space to store these pathology slides. So if you can digitizing these pathology slides, you can ensure the images will have not degraded over time comparing to the physical slides. So this is one of the advantage to digitize the pathology slides. And there are several slide scanner to do this job, and it's also approved by the FDA to scan all images, because you need to make sure the scanned images is comparable quality to the microscope. Okay, so this is the one of the technical barriers need to be broken, break in the pathology, digital pathologies. Okay, so for example, here you need to make sure the image quality can see the very small things, even in the 3 micrometers. And so this is called the whole slide imaging, and there are several companies to already develop the digital whole slide imaging machines to help you to digitize everything. And for these machines, now you probably need to know that so if you have experience using scanners, then it probably takes a little bit time to finish one scan of the paper. But in the pathology slides, you just imagine that you need to use microscope to scan everything in these slides and in a very high detail. So for even for the one slice, it probably will take lots of time to finish one slide scanning. So the development of these whole slide scanning images, the machines, is actually need to be very fast. So for example, if you are in a very large hospital, then probably will have more than hundreds of the pathology slides need to be read every day, even thousands of slides. Then if the machine can only scan one slide for like even for 5 minutes, then it's still not enough to implement this digital pathology into the clinical practice, because you need a really fast machine to scan these slides. So, which means you need an automated, high-speed, high-resolution whole slide imaging system to do this kind of job. Okay, so assume that you already digitized the pathology slides, and the next thing is that you need to do some machine learning analysis. And this is very important because not only you can apply some algorithm to identify the abnormal tissues, then the digitalization of the pathology images will also enable the telepathology to let the doctors in a remote hospital can consult the pathology images with another doctors in another region. So this is one of example published in China, that they have a percentage of utilization of the digital consultations about the pathology images. So that is very one of the advantage of digital pathology that change the practice of the pathology, because in the older time, if you want to consult the pathology image with another pathologist, then you need to mail the slides to the other place, and to take lots of risk, because the slides may be lost or just may be broken. So the telepathology is one of the advantages of the digital pathologies. But in terms of the machine learning, that I just want to show you one of the very interesting paper published in 2017, that it is the researcher, they just try to look at if the Google image search can identify the benign or the cancer tumor. So in this research, they just try to upload the cancer images onto the Google image search, and they find that for the benign tumor, they have 83% of the rate that can return means that 83% of the images shown in the Google image search is also benign. But for the tumor, it's like 50%. So it's just like that, okay, even for the Google image search, it has some basic mechanisms of pattern recognition, can also do the job to differentiate the tumor and benign and the cancer. And so there are lots of the development in the digital pathologies. For example, you can develop a software to do the whole slide scanning and to visualize these whole slides on your computers. You can magnify these regions and you can see if there's a match of certain regions to these patterns, and you can use some computer algorithm to do this kind of job. And for example, you can also develop an AI algorithm to identify the cancer cells in the whole image, because not every cell in the pathology image will be cancerous. So you need to label these cancer cells using the AI algorithms based on the heat map. So this heat map can help pathologists to not to miss any cancer regions. And for example, Google has used AI to detect lymph nodes metastatic breast carcinoma and published in a very prestigious papers. And also the AI can help for the mitosis detection, and it can detect the very subtle nucleus images to see if this is mitosis or the normal images. And also you can try to count different cells based on the different immunohistochemistry, the label of the cells. Then there was a paper showing that using a deep learning convolutional neural network can recognize common patterns of injury in gastric pathologies. So it just try to identify the pathology images from the endoscope scan, endoscope biopsies, and it has achieved very high accuracy. So I think it's too good to be true, but it's just one of the results of from the neural network. And in one of the papers in the Nature Medicine, it published 2 years ago, it shows the classification and mutation prediction from non-small cell lung cancer histopathology image using deep learning. So it has also achieved quite a good accuracies, ranging from the 0.73 to 0.856. So because the reason that this paper published in the Nature Medicine, because it can be applied to any cancer type, and it has also a published code for this kind of job. So it's one of the pioneer paper in the digital pathology. And there are lots of the startup companies try to develop an AI algorithm or AI platform to diagnose the pathology images. And this is one of the first FDA-approved AI company to provide the digital pathology AI diagnosis. And it's also one of the demo figures that showing that they are able to use the heat map to identify the abnormal tissues in the pathology slides. And also, one of the paper also shows that the digital pathology can actually save your money. Okay, so imagine that in the hospital, you need to purchase the slide scanners and also to purchase some the computers to instead of the purchase, just purchase the microscope. So in this paper, actually show the digital pathology can have a 12 to 30% efficiency gain at the pathologist level, and you also save on the space for these pathology slides. Okay, but for the digital pathology, we also need to think that the overall the cost and effect of these transitions. And in Taiwan, and also there was a very famous company called Yunxiang Keji, and they are also devoted to the pathology image analysis and to provide these AI tools for imaging analysis. So I think in the first part of the lecture, I just want to give you a flavor of the pathology image analysis and how this image analysis can transform the traditional pathologists like this one to using microscope to look at the tissues. From this one, you can just use the computers to look at the images. And I think that is also kind of the trend in the smart hospital, because the microscope, although it's very important, but sometimes it just is like a burden to pathologists, because almost there's a high rate of the retinal pathology, retinal problems for the pathologist. But if you are using computers, probably it could be safe to help you to prevent from your eyes to have some retinal disease. Okay, so for the future of digital pathologies, I think that the advantages are the efficiency and accuracy, and we can preserve the image qualities, and also for the easy for sharing and consultations and eyes protections. But there are some limitations, because in the current digital pathology, the workflow is not entirely automated, and you can imagine that some steps are not easy to automate it. So if you are interested, you can consult for a pathologist, and he or she will tell you which parts of the workflow cannot be automated. But that is the limitation you need to overcome. And in the digital pathology, there are still no standard for the image formats. So this needs to be overcome. We need to unify the image formats. And the overall, we need to evaluate whether the digital pathology can really improve our efficiency, or we actually do more work because we just need to digitize everything. Okay, so, but based on these lectures, I just want to show you a workshop that how to apply a convolutional neural network to differentiate the tumor, benign tumor and the cancer cells. So just let's take a rest and move on to the workshop parts. Thank you very much.